Here in part 2, we'll focus on morphological diversity in archaea and bacteria. Key to understanding the morphology and structure is to recognize that both archaea and bacteria have a cell wall that surrounds the entire organism. This is a hard, rigid wall that prevents the cell from expanding too much in hypotonic situations when the cells might otherwise expand. Inside of the cell wall is a membrane, and within the membrane we'll find the cytoplasm and the nucleoid. They lack a distinct membrane-bound organelle that contains the DNA. Instead, it's in a nucleoid. Prokaryotes, archaea and bacteria, also have a flagella for locomotion and pili surrounding on the outside that can serve sensory functions and also are useful for the exchange of genetic information through conjugation. Archaea and bacteria are also able to form a capsule of different kinds, which is a protective structure which allows archaea and bacteria to persist dormantly through very difficult times to resume life when resources become more available or more beneficial, conditions more beneficial. So what are microbes? It's a generic term that we use to describe archaea and bacteria, but it's also applied to describe some eukaryotic organisms, protozoa, fungi, slime molds, algae, even amoeba. We'll be talking about some of these later. In particular, in 3 lectures, we'll be talking about fungi. But to say that it's single-celled is a bit of a misnomer, because sometimes these single-celled organisms can link together, such as you see here for cyanobacteria, to form a string, where the cells perform different functions. You have some cells, called vegetative cells, that are conducting photosynthesis, whereas other cells have a stronger cyst or larger cyst around them, and they perform a nitrogen reduction function, taking atmospheric nitrogen and producing ammonium. We'll talk about this in the next lecture on carbon and nitrogen cycles. But the point here is that these single-celled organisms can form communities. So at this point here, we're going to stop and have a clicker question on a 2-word phrase describing a microbe. Bacteria and archaea exhibit tremendous morphological diversity. They can be as small as 0.3 micrometers, such as you see here for mycoplasma, or 300 times larger, as you see for Margarita. They come in a variety of shapes. Some are circular, some look bead-like, some are rods, some look like they have a spiral shape. There's a variety of shapes, as well as a variety of mobility. Some are not very mobile. Others have a long set of flagella that are used for locomotion. Others glide through the environment, such as you see here for Oscillatoria. And they live in extremely different environments, from as low as 5,000 feet with high pressures below the ocean surface, to very high temperature, low pH environments, such as you might find in Yellowstone National Park, to very complex systems, such as the gut of an animal system. Here are 3 examples of extreme prokaryotic life. Geogemma, from the hydrothermal vents, lives at 121 degrees C. That's hot enough that they can grow inside an autoclave. We have archaea that live in very, very harsh environments, such as the Great Salt Lake, where organisms are living in essentially a saturated salt solution. Or we have organisms such as Deinococcus that can persist in environments with very high ionizing radiation. When we begin to consider the diversity of pH environments that are found on our planet, from very low pHs to very high pHs, we will find archaea and bacteria. We call those that live in a very high pH environment alkaliphiles, and those that live in very low pH environments acidophiles, with, of course, those in neutral pHs being known as neutrophiles. This table here simply summarizes some of these extreme conditions, providing examples, so that we see that there are organisms that persist in ice to temperatures above 100 degrees C, across the range of pHs, across the range of ionizing and UV radiation, high pressure, high salinity. Essentially, all parts of our planet have archaea and bacteria. If we begin to look at the organization of archaea and bacteria on the tree of life, we start off here with Leuca. What we find is that there's a branching point. Bacteria are found on one branch, and archaea on a second branch. And it's also that on this branch we find the eukarya that we'll be talking about in 2 lectures. We use the term prokaryote to describe the characteristics of archaea and bacteria, these 2 groups. They lack a distinct nucleus with a membrane, and they lack specialized organelles.